Here's a thing that they talked about in Grossbeck's class, but not Baker's. Okay, oxaloacetate. Do you remember him talking about that? He talked about a 4-carbon chain called oxaloacetate in the Krebs cycle. Okay, so we're in the Kreb or citric acid cycle. The Krebs cycle? Krebs cycle. Kreb. No apostrophe S. What am I thinking?
Krebs cycle.
Okay. Just so you know, in your class they talked about something called oxaloacetate. I'm not 100% sure if you need to know it for the test because the other teachers I don't think have covered it. But so there's a 4-carbon chain called oxalo.
But he mentioned that in Brother Baker's class.
He did?
Mm-hmm. Because
Oxaloacetate.
it attaches with acetyl-CoA,
Yeah,
doesn't it?
I didn't know he talked about it
Don't
Baker's. Sorry about that.
Maybe he didn't, I just got it from the other tutor.
I
But
do remember oxaloacetate.
okay, so oxaloacetate is a 4-carbon chain, and it's going around the Krebs cycle all the time. It attaches to the 2-carbon acetyl-CoA, okay? And it's going to take it all the way through. We're going to use up the 2 carbons from the acetyl-CoA, and we're going to be left with the oxaloacetate at the end, which is going to grab another acetyl-CoA, go through it again, and just keep going. Does that make sense?
I'm still not sure where the oxaloacetate
Oxaloacetate
came from.
It's
So-
just in the.
It's just in it?
Yeah, it's just always in there. It's just,
Oh.
Kind of like
it's just,
that.
it's just,
Just
a
one there?
Just chilling there, guys, saying what's up.
So oxaloacetate resides within the matrix of the mitochondria.
Okay.
And so it's attracted to acetyl-CoA specifically. So that's every time there's an acetyl-CoA available, it's going to grab acetyl-CoA, carry it through the Krebs cycle, and it uses up the acetyl-CoA. Then we're going to find another acetyl-CoA if any is available
Okay.
and continue in the Krebs cycle. Okay? Okay. So real quick, just so you know, from this point forward, I've been talking about one pyruvic acid going in. So all these numbers would be doubled. Okay?
because there's 2.
If they asked you in the Krebs cycle, what are the products from one glucose molecule? Or if they asked you for one pyruvate molecule, then this is what it would be.
So it would be doubled just because of the 2.
Because if both
So if-
go, yeah.
Do they always both have the same thing? Like they both have oxygen or they both don't have oxygen?
Think of it like such. They both are going to have the same fate. Okay?
Okay, sounds good.
Okay. So let's think of the products that we're going to get. I just told you that the carbon's going to be used up. So how did we use carbon before? How did we get rid of carbons?
Good phosphates. Or
I think
we haven't gotten
that's
rid of any.
split. We got rid of some because this is a 2-carbon chain and it used to be 3.
Oh, that's right, we had carbon, oxygen.
Oxygen. So we're going to get rid of 2. So we're going to have 2 O2 molecules come in. We're going to get 2 CO2. Okay? That's your question face, I know it.
Sadie knows that already.
Because, sorry.
What about the CoA? I thought they grabbed the carbon.
Okay, I'll answer that in 2
But
seconds.
doesn't one of them knock off? One of the carbons knocks off, right? And that's how it makes the carbon dioxide.
Mhm. Okay, so CoA is attached to this, okay?
Okay.
So
it's
we
not a
have
part of it.
acetyl-CoA.
Okay.
Acetyl-CoA attaches to oxaloacetate.
Yes.
So now it's a 6-carbon chain. It's citric acid or citrate. Yeah, I think I said it right.
Okay.
Then you're going to have one oxygen molecule come and it rips off a carbon. So it's a 5-carbon molecule.
Mhm.
Turns into another name that I'm not going to go through. Then another oxygen, O2, comes by, rips off another carbon molecule.
Okay.
And you're left with
So now we're down to
4. It's not technically called oxaloacetate yet, but it's the 4 carbons from oxaloacetate that are left over.
Okay.
So now we have a 4-carbon molecule with CoA attached to it.
Or is CoA
I'm not
part of
100% sure, to be honest. I don't remember if CoA gets detached before or what.
Okay.
I
Probably doesn't
I
matter.
know it's a lot more. actually, when you guys take Bio 180, they'll talk about this way more in depth. Luckily, you don't need to know it way more in depth right now. So I won't torture you.
Okay.
What are gonna be the other products?
NADH.
So we're gonna have NAD plus come in. And we're gonna form NADH. How many NADH?
Just 3.
3.
3.
So 3 are gonna come in. We're gonna get 3 NADH. Is that alright?
Mm-hmm.
Just remember these because these are the products, okay? And what else?
FAD.
FADH comes in, and we get FADH2.
Oh, FAD comes in and we get FADH too.
It's FADH.
Oh, really?
Yeah. I've seen someone put it as FAD plus before, but more frequently I've seen it as FADH and turns them into FADH2. So if I'm wrong on that, I do apologize, but from my best understanding, it is FADH that gets turned into FADH2. Okay? One other product. One other product.
ATP.
ATP. So this is 1, then 1 ADP comes in, and we get 1 ATP. Looks like a lot. Feels like a lot. There are 4 things. We get CO2, NADH, FADH2, and ATP. Just 4 things.
You have to times that by 2 because it turns twice.
Times it by 2 if you're talking about a glucose molecule. Okay?
So instead of 3 NADHs, it's 6 NADHs?
Yeah, it would be 6 NADH, 4 CO2, 2 FADH2, 2 ATP.
Okay.
And you would also do that to these guys up here, so 2 CO2 up here and 2 NADH up here.
When do you times it by 10?
If you're talking about a full glucose molecule
Okay.
versus a pyruvate molecule. Does that make sense?
Yeah.
I know it's a bit,
A lot
you have
more.
to review
Yeah,
it.
definitely. Just look at it.
Yeah. 4 products, looks like a ton. In the Krebs cycle, 4 products. Don't forget the transition phase right here, though,
Yeah.
where there are 3
The
things.
CO2 and the
CO2, NADH, and then CoA comes in.
Okay.
So 3 things happen here. Under it, 4 products. Okay?
Okay.
And if you know the product, you usually know what's coming in before it. Before NADH was NAD+. Okay?